Many groups around the world are working on ways of increasing the efficiency of meat production. One of the potential solutions is cultured or lab-grown meats, which attempts to create meat and animal protein products from isolated biopsies of animals. This sidesteps many of the ethical issues associated with conventional meat, but it is primarily being investigated for an increase in efficiency. Meat production is one of the most resource-intensive areas in food production, in water, land, and energy. As well as the current burden that this industry puts on the environment, there is also the expected increase of the human population and demand for meat among people. New strategies are necessary to make meeting this demand even feasible, let alone sustainable. There have been successes in culturing a sufficient amount of animal muscle cells to create an edible product. These small amounts of edible cultured meat have been extremely expensive to produce, but it's only a matter of time before costs go down as the techniques are refined. Still, there are many challenges in this area of research. Cost is one of them, as well as finding a suitably efficient growth medium to maintain efficiency, replicating the proper taste, and giving the product texture. Today we're going to focus on the issue of texture, as it is the challenge with the least obvious solution. Many ideas for efficiency are already proposed, mainly in the form of growth medium derived from highly efficient cyanobacteria. Taste can also be easily improved from a conceptual level by culturing fat cells with muscle cells, along with adding certain specific metabolites to the growth medium. Providing proper texture requires replicating the extracellular matrix, as well as larger strands of connective tissue within the piece of meat. The stem cells responsible for the ECM and connective tissue can obviously be cultured alongside muscle and fat cells, but getting them to behave and develop into the proper structure is another issue entirely. This is the same issue raised in medical tissue and organ engineering, but their solutions are likely not going to be suitable for a food product, especially for complex organs. One strategy is to 3D print the cells layer by layer so that they form the proper structure. However, this is extremely time-consuming and not appropriate for the scale necessary with industrial meat production. Another option is artificial scaffolding spun from biocompatible polymers. This provides several technical and perceptual challenges. Even if such polymers were completely safe to consume, consumers may not feel comfortable eating something that did technically contain plastic. As with the 3D printing, another issue is the time and complexity necessary to actually form these initial scaffolds that the cultured cells would then grow on. To address this biological challenge requires a biological solution. To our knowledge, no group is currently investigating anything like this. It is merely a concept, but all of the ideas are supported by existing knowledge and research that is referenced in the video description. The idea is to establish a preliminary extracellular matrix for cultured meat production, starting with a genetically engineered fungus. This idea starts from the superficial structural similarity between the extracellular matrix in animal tissue and the filaments of fungal mycelium. To serve as an appropriate foundation for animal muscle tissue, the fungus would need two primary modifications. One, the production of certain animal extracellular matrix proteins, and two, a programmed kill switch. From a conceptual perspective, both of these goals are feasible. There has already been successful genetic engineering of multicellular fungus using the latest genome editing tools. Likely, the most challenging part of this idea would be getting the outer surface of mycelium to chemically resemble animal extracellular matrix. The cell wall of fungi is primarily made of the structural polysaccharide called chitin, with a small amount support proteins. A key first step would be downregulating the production of chitin and increasing the percentage of cell wall composed of protein. Next would be adding to or replacing fungal structural proteins with animal collagen. The synthesis pathway of collagen is complex, but modifying organisms by inserting multiple genes is not unheard of. While certainly a challenge, both collagen and fungal structural proteins are exported from the cell in a similar fashion. Once the challenge was met, it is possible that the foreign proteins would be readily integrated into the cell wall. It may also be necessary to express other forms of animal structural proteins, but research will be necessary to determine that. The second modification is a programmable kill switch. Scientists have already developed a highly efficient and sensitive gene circuit that allows for programmed cell death with the addition of a normally harmless substance into the growth medium. Once the modified fungus had grown sufficiently so that its mycelium provided a suitable scaffold for animal tissue, the kill switch would be initiated. This would prevent the fungus from draining any further nutrients from the growth medium and competing with the animal tissue. and it would leave behind simply the structural threads of the mycelium, ideally composed primarily of animal collagen. From there, the scaffolding would be seeded with animal stem cells, including those associated with muscle, fat, connective tissue, and perhaps even vasculature. The final texture of the product would depend on how efficiently the fungus can be made to express collagen, as well as how much initial scaffolding is necessary to start the process. It may be that the fungus only needs to provide a very loose structure before animal stem cells can take hold. This idea may sound very complex, but it may be the best solution. It is possible that animal cells that normally produce extracellular matrix could be coerced to produce such structures on their own. However, the establishment of connective tissue is always formed alongside all of the other cell types in a tissue. The purpose of the modified fungus is to grow these structures en masse and very efficiently. The mycelium naturally produce these kind of structures in growth medium, and it may even be possible to modify the growing condition or genetics to get different structures and therefore final textures. Once established, the fungus-based scaffolding can then be sectioned off and used to grow a wide variety of cultured meat products, making highly efficient cultured meat a viable and appealing replacement for many everyday uses. We hope you found this idea and video interesting. If you did, please share this on social media, and especially if you know anyone interested in or related to cultured meat research. Again, this is just a scientific idea, but maybe someone out there can make it a reality. Thank you for watching.